I will start introducing Dr. Glazier. I'm delighted to invite you all to the 9th IBSE Colloquium. So we've had a lot of eminent speakers, and the 9th in this line of speakers is Dr. James Glazier. And it's wonderful to have him here. Thank you again for accepting our invitation. He received his BA in Physics and Math from Harvard University in 1983, and his MS and PhD in Physics from the University of Chicago in 87 and 89. So he's a physicist by training, and he's held faculty positions in a number of universities, including UCSB and in Western Australia and in France and in Japan. So he's been quite a globetrotter, even on academic positions. His research focuses on applying physics-based computer simulations to understand various aspects of development, embryonic development, toxicity, and also developmental and infectious diseases. And he applies simulations to develop and optimize disease treatment. He's an active member of several interesting consortia. Particularly, he leads the CompuCell 3D multiscale modeling environment. Some of you may have already used it, and also Mechanica. I'll share the links. You can check them out if you haven't already. And he also teaches an annual week-long workshop on multiscale virtual tissue modeling. So I think Sheriff already shared those links with all the participants of our school, but I know there are others who are here, so we will share those links again in the chat for your benefit. And in 2016, he became one of the founding members of Indiana University's Department of Intelligent Systems Engineering, basically trying to apply advanced computing techniques to understand and control engineered emergent systems. He's also a fellow of the American Physical Society and Institute of Physics in London, and he co-leads the IMAG MSM working group on multiscale modeling. Can also share that link. It's a very nice initiative that's nucleated during the pandemic and is going great. So with that, I'll also hand it over to Sheriff, who can say a few more words because he's had more of a personal association with Dr. Glazier, and then the floor will be to Glazier for his talk. Sheriff.
Thanks, Karthik. I think you already covered most of the biography of James. I think one thing I would like to quickly mention is about the last group, the IMAG multiscale modeling group, where I'm also involved in. That's where I met James, and we have regular meetings there. And then one reason I go there is because I always get to learn a lot from the discussion there, and one of the main contributors of the discussion is James. Yeah, every time I go, there's always something new I learn. I learned a lot about digital twin from James' discussion, and he has very good clarity on this, and he's working on those areas and on online and offline digital things. I think I haven't heard the explanation for the digital twin better than anyone, no one has given better explanation on these topics than James so far from everyone I have heard. I think he's really a pioneer in this field, and he's indeed working on this CompuCell 3D, which is also kind of very useful, interesting modeling tool for building multicellular tissue models. I guess you will learn a lot about this in this session. Overall, he's very good in articulation. I'm sure all those here will definitely benefit from this talk. And thanks, James, for accepting to speak in this webinar, as well as which is also part of our winter school, IIT Madras EBI co-hosted winter school. It was nice to have you. Looking forward to your talk.
Thank you very much.
Thanks, Sheriff. Over to you, Professor James now, and request the participants, please post your questions in the Q&A, and you can also upvote questions so that it will help us pick out the more important questions or the most wanted questions for Professor James to answer. Over to you.
Okay, thank you very much. I'll try not to take too much of your time. I appreciate you being here. The work I'm going to be talking about today has been done by many, many people over the years. I've tried to list most of them on the cover sheet. You'll see a lot of names from a lot of institutions, because this has been highly collaborative work in a variety of areas. And what I'm going to give you today is just a very high-level flyby of a bunch of projects, any one of which could be dug into for a longer talk. If people are interested, please contact me about details. Maybe it's worth saying just a word about Indiana University. Indiana University is in the middle of the country, pretty much. It's the southern part of Indiana. I sit in the Department of Intelligent Systems Engineering, which, as you heard, was established in 2016, so we're a new program. We are part of the Luddy School of Informatics, Computing, and Engineering, which does a lot of training in the area of data science and machine learning. It's a popular set of programs. The idea of ISE was to take computing and apply it to complex engineering problems in a variety of fields, ranging from neuroengineering through cyber-physical systems to bioengineering. And I guess would represent the bioengineering component of that. And we have programs at undergraduate, bachelor's, and master's, combined programs, master's degrees, and PhD. If you want to find out more about the program, you can see it engineering.indiana.edu. We have a large contingent of students from India, and we are always happy to talk to you about your interests. We come to the problem we're talking about today, and I'm sure you've heard about this a lot in the rest of the workshops, because medicine is not designed in the same way that other engineered systems are designed. Computing is not really a fundamental component of most medicine, medical practice, or therapy discovery yet. And one of the problems with that is it means that we don't have a true personalized medicine. We don't have the ability to determine what optimal treatments are, because we can't reproduce experiments. You can't operate on somebody and not operate on someone at the same time. It's not really scientific because you can't do control experiments. You see that we can't always predict which treatments will work, and therefore we reject treatments. And medical care is still given in what we'd call an open loop fashion. Medical care is responsive or reactive rather than proactive. There's a question for sharing the link, so I guess we can do that. And we need to be able to understand better how to prevent major problems or intervene when problems are minor, rather than waiting for catastrophes. And certainly the idea of digital twins coming from engineering, especially from the design of airplane engines, where GE was a pioneer in this area. The whole point of a digital twin is to allow two things, one of which is to be able offline, to be able to try out different strategies for treatment and come up with optimal solutions. The other one is to run a digital twin in real time as systems are running and look for deviations from expected behaviors in order to be able to predict failures. And that's certainly been the main reason that airplanes have become very safe over the past 30 years. Doing that in humans is very difficult. We're not going to get there today by any means. The things I'm going to talk about are more basic science and offline digital twin type ideas. One of the results of that push for more sophisticated computing and mathematical informed medicine is that while we've heard a lot about the need for data scientists and artificial intelligence machine learning experts, under the rug, hidden, there's a huge demand for people with the kind of mechanistic modeling experience that you're going to get in this course or that we're going to talk about today. We can point to some of the reasons for that. The U.S. Food and Drug Administration now requires that you do pharmacokinetic, pharmacodynamic models, so-called PBPK models for new drugs. And as a result of that, all the big pharma companies are hiring people in the area of mechanistic modeling, the kind that this course would deal with very much. There are new companies and we could point to a couple of them. HeartFlow Diagnostic Company out of Stanford, Applied Biomath in Boston, Nova Discovery in Europe, in Lyon, which are doing diagnostics, custom mechanistic models, clinical trials, using mechanistic models. Here in Indiana, there's a new Embryo Institute. It's an NSF-funded institute, which is trying to greatly expand the number of PhD students, in particular, and postdocs in the area of mechanistic modeling. Indiana University, my university, is a participant. So is Notre Dame, which is one of the other big research universities in Indiana. There are new initiatives throughout the United States from the federal agencies, National Science Foundation in particular, but also private foundations, Simmons Foundation, Chan Zuckerberg. As a result, there's a major shortage of people with training in mechanistic modeling of the kind that this workshop has been working on. And it's never been easier to get an academic level postdoctoral position or faculty position. And I think I'm mentioning this because I know there are students in the room. I think everybody is aware of the need for AI and ML, data science, but I think people may not be so aware that there's a shortage of people doing mechanistic modeling. So if we want to be able to predict, and ultimately in order to be able to control things as engineers or scientists, we need to be able to predict. We need to understand how they work. And my work over the past 25, 30 years has been focused on trying to understand the mechanisms of development. How do you build an organism, or how do organisms build themselves? Homeostasis, how do organisms maintain themselves? What we would call developmental diseases, that is, how do those homeostatic maintenance mechanisms fail and lead to disease? And then more recently, asking questions about infectious diseases and how they work, how we can control these processes in bioengineering, and how these processes can be disturbed in toxicology from chemical and other insults. All of these exhibit very complex interplay between physical and biochemical mechanisms. I think you've heard a lot about biochemical mechanisms in this workshop. Today, we're going to focus a little bit more on the biophysical side of things. If we look at embryonic development in particular, one of the things that we see is that there's a lot of movement. Cells move, they change shape, they change their relationships to their neighbors, they build structures, and understanding that kind of structural reorganization and how it interacts with the signaling between cells and within cells has been the core of what I do. And all of the examples I'm going to show today are related to this kind of question. When we think about these kind of biological problems, one of the things we face computationally is difficult is the range of scales that are involved. Everything from single molecule interactions in metabolism, higher scale signaling mechanisms between and within cells, the organization of cells into local architectures like capillaries or small regions of tissue, the organ systems of the body, and then the interactions of the organ systems. Each one of these may have its own way of modeling, and we have to think about how to integrate them. We tend to think of that, call that integration virtual tissues, and typically we do it at 3 or 4 scales. We'll have the kind of network models that I think you've been hearing about today in the past couple of weeks, right, in much detail, modeling what's going on inside of cells. That will control a layer where we have cells and their behaviors. Those cells and their behaviors will be in an environment which could have extracellular chemical species, extracellular matrix, and other boundary species. And then that will often sit nowadays in our work inside another ODE, non-spatial model, pharmacokinetic, pharmacodynamic model, which would modeled the blood flow and the distribution of molecular species within the whole body. Typically, we focused on the tissue level. That is, how do we organize a tissue? We'll come back to this picture. This is of embryonic development, of the formation of the segmental pattern of the vertebrate spinal column. Ultimately, this could be used, if we can do it right, and we're still relatively far from that, to take the kind of static imaging data that's being generated by the omics revolution, the spatial omics revolution, and animate it to give you living movies of how tissues evolve. If we're going to describe tissues, one of the things that we have to be able to do is understand how cells interact and how they move. And a lot of the work that I've done over the past 30 years has been focused on this kind of biophysical modeling of cell-cell interactions. If we're going to describe the dynamics of these patterns, we have to know where the components are, what the components are, how they interact with each other, both in terms of force generation and in terms of signaling, and how they respond. And so we've developed over the years a number of ways of doing that. We use something called GGH-CPM, which is a lattice-based method for describing cells. In a sense, it's a very naive method. If you imagine taking a digital photograph of a group of cells, and you say that all the voxels or pixels in cell number 1 are labeled 1, all the voxels or pixels in cell number 2 are labeled 2, that's how we do it. That's a lattice-based method. There are models which model each cell individually as a point particle. Those are called center models. Molecular dynamics models in computational chemistry are a classic example of that. And there are other species of models as well. Immersed boundary models come out of more finite element solids modeling approaches. You can cluster multiple center model particles together to make subcellular element models. There are many different ways of modeling these. One of the hats that I wear is a software developer. You've heard mentioned CompuCell 3D. It's a virtual tissue simulation environment with an editor and an execution window. These allow you to build relatively compact Python specifications of models. If you want to try it out, there are downloadable versions available from www.compuscell3d.org. If you want to try to run it yourself, maybe during the class, so to speak, and you don't want to download it to your computer, there's an online version, nanohub.org/tools, CC3D base 4X, because it's version 4. You have to register for NanoHub, but it's free. Anybody in the world can use their servers and run code on it. This is a picture of the NanoHub version. It's CompuCell 3D webpage, not so pretty. And this is what the editor looks like and the execution. All of the simulations I'll show today were written in CompuCell. CompuCell, as I say, is a scripting environment. The ideas in CompuCell are to try to provide primitive functions that are natural in biology. The idea of chemical fields, extracellular matrix fields, and what they do: diffusion, decay, secretion, absorption. Cells, we call them generalized cells because they could represent also subcomponents of cells or components of non-cellular materials, which have volume, surface area, elasticity, motility. Cells can grow, they can divide, they can die, and they can interact with other things besides cells, like chemical field. Solids modeling in a basic way, links, triangulations, elastic moduli, and then again tying back to, I think, the main theme of what you've been doing here, which is dynamic networks. And so one of the things that's most interesting is when you run a signaling or metabolic network or regulatory network by itself, it tells you a lot. but those networks aren't operating inside a vacuum. They're not operating by themselves. All of the cells in a tissue are interacting with each other, and so the signals for a network are continually changing. And one of the things that CompuCell allows you to do, and I'll come back to this, is you can put network models written in SBML, MABOS, or other standards inside of CompuCell cells and see how that changes the behavior of the network. CompuCell is designed to be easy to use. If we had a little bit more time, I'd walk you through building a CompuCell simulation. You can literally do it in 60 seconds. There's a little bit of a simulation templating wizard that allows you to define the components that you need, assemble them through a set of menu dialogues, and build a simulation. The code itself that's generated looks a little bit like this. You have some XML, CC3D-ML, which is a simple markup language that specifies static properties of a model, like how many cells you have of various kinds, not the number of cells, but how many species of cells, what things they are capable of doing, and then dynamics are written in simple Python scripts. If you want to change the size of a cell, if you want the cell to appear or disappear, if you want to provide graphics outputs, and so on. And this is a typical simulation. This is the cell sorting simulation, which is sort of the basic, if you're a physicist, the hydrogen atom of this kind of modeling. And it allows you to then view it in various ways. I mentioned before, and this is a little bit out of order, but because of, I think, the background that you have here, I thought it would be important that you can put network models inside of cells, inside of environment, and those allow you to run them in a whole variety of ways. Here's an example where we've taken a cell cycle model, a famous Talsisin cell cycle model. You can grab it from the Biomodels database, download it as an SBML model, attach it to individual cells, and control the cell cycle inside those cells using that model. And it only takes a couple of lines of code to implement that. Here's an example using the antimony model network specification language of what's called delta notch signaling. In this case, there's a signal not only within cells, but between cells. Cells have a pair of signaling molecules, a delta on delta and notch. The delta is essentially sending a signal. Notch is a receptor. When the notch signal is received, it sends a signal to the cell to differentiate in a particular way. Here you can use simple text-based specification, antimony, which can be converted into SBML very easily, and build a model of this kind. Here's an example of what that looks like when you run it, and this is the full code that's required to build this simulation of cells interacting through contact between their neighbors. Our most recent extension was MABOS. A lot of people don't like to use these reaction kinetic models because they have too many parameters. Many people use what are called stochastic Boolean models, which have fewer parameters to specify network behavior. This is that same delta notch signaling between cells put into individual cells, and that delta notch actually controls a cell cycle model. Not quite the one I showed before, but very similar. And here's an example of that simulation running. And here's the code for that simulation. So now I'm going to spend the remaining time that we have together giving you just a few examples of the kinds of problems you can solve or address using these kinds of modeling tools. I promised something on Drosophila wing patterning. I only have one slide on it, but I think it's a very beautiful example. This is another case in early embryonic development in Drosophila. The cells have hairs on them. These are not the hairs on your head because those are multicellular. These are fibers on individual cells. Those fibers have an orientation. That orientation is determined by the movement of molecular species within cells due to the interactions between cells. And what happens in this case is that at the boundary of the tissue, there's a signal that's sent. It causes orientation of molecular species within the cell. That signal then is transduced through the tissue and spreads to give an oriented axis for the cell of planar polarity. The particular reason that I show this is because it actually shows this idea of a generalized cell, which we'll come back to. Here we're actually modeling the specific molecular species within the cell and how they move. So the CompuCell cell typically is a blob, a single object, but here we've actually subdivided into compartments which move, which represent the movement of molecular species within the cell, giving rise to the polarization and the orientation of the spikes in the cells. Another example where we use this kind of idea is modeling segmentation. This is work we've done over many years. I showed this picture before. In the early embryo, you have two streaks of tissue along the midline of the embryo. These are called the presemitic mesoderm. Initially, they form basically a solid column. That column reorganizes to form these little ball-like structures, which are known as somites. The way that that happens has been studied for many, many years. They form sequentially. This is a movie of a chicken embryo forming the somites. You'll see the spots appearing sequentially. You'll see the presemitic mesoderm form, and then it organizes into these ball-like structures. Those structures will then go on to organize the vertebra in the embryo. If you're a snake, that process takes a long time because you have hundreds of vertebrae. If you're a fish, it happens pretty quickly. We have fewer. And that's a big way that the body plan of the embryo is created. There's a long history of modeling of this kind of system. In particular, there are oscillatory genes within the individual cells. Those oscillatory genes interact with differentiation signals, and that differentiation signal is read out essentially because of the phase of the oscillator at the time of differentiation, and turned into expression of structural proteins that determine what the cells do. And over the years, and this is the same movie we saw earlier in a little more detail, we've built a series of models trying to understand how these molecular signals translate into changes of cell behavior that lead to structural changes in the tissue. On the one side here, you will see the oscillatory genes within the cells. Here is a gradient of a signal that's going to induce differentiation. On the left-hand side is the differentiation state, which leads to mechanical changes and then to segmentation. One of the things that we were very interested in is that this is quite a robust mechanism. Even if you mess up the initial condition, you'll get a few bad somites, and then you reorganize the structure to form the correct pattern. We did some experimental work with colleagues in England. The work was done in the laboratory of Claudius Stern, University College London, and he discovered, rather surprisingly, that you could turn off the oscillator and the somites still formed. And so one of my graduate students then built a model which was a mechanical model of semitogenesis. In this case, our cells are now modeled as extended objects with what's called a basal and an apical region. I've actually put that backwards. The red is basal, the green is apical. And we could model then a mechanical process that led to segmentation as well. You could ask, if you have the ability to build segments mechanically, why do it through this complicated signaling and chemical oscillatory network? The answer is that the mechanical mechanism does pretty well, but it's not very accurate. And if you put the two things together, you have a much higher accuracy of the segmentation. Another thing that we've worked on now, jumping from development to disease, is called choroidal neovascularization. This is age-related macular degeneration. Normally, the capillaries that supply your retina there's a thin layer of them on the front, there's a layer of them in the back. In especially the elderly, those capillaries in the back can cross into the retina, invade the retina, and begin to destroy it. And we were trying to understand why that happens, or perhaps the better, why it doesn't happen in most people, at least until they're 85 or 90 years old. And the answer that we came up with was not the one that was in the medical textbooks. The medical textbooks would have said that the problem was excess of a growth factor. However, there were particular observations that in different patients, the blood vessels spread in different regions of the retina. They were localized to particular regions of the retina. That wouldn't happen with the growth factor, which is distributed everywhere. And so we built some series of simulations that focused on possible mechanical defects leading to blood vessel invasion and disruption of retinal structure. And I always love showing this movie. I think it's one of the most beautiful movies ever made with CompuCell. Here you have in the back the blood vessels in purple, in blue-gray what's called Brooks membrane, which is a non-cellular structure that separates the blood vessels from the retina. In green, the black cells that line the eye, the pigmented retinal epithelial cells. And here in gray, the photoreceptors, which are actually doing the sensing in your retina. If you make a small hole in Brooks membrane, and adhesion is normal, and by the way, these holes happen all the time in normal life, the blood vessels don't invade. If there are defects in the adhesion between cells, the blood vessels are able to invade. They cause retinal detachment, which you see here. They proliferate, and eventually they'll cause the breakup and loss of the photoreceptors. One of the things that we were very proud of with this simulation was that the time scale predicted for the disease was about right. You lose your vision in about a year after this starts. We were able to take that and make a more medically significant prediction, which was to say, what's the risk of particular forms of this disease happening as a function of particular cell-level pathologies? You could say, if there were defects in adhesion within the pigmented retinal cells, what happened? If there were defects in adhesion between the pigmented retinal cells and the photoreceptors, what would happen? And in this case, if there was a defect in adhesion between the pigmented retinal cells and that membrane underneath them, what would happen? That leads to the classic form of age-related macular degeneration. And it happens also that lipids accumulate inevitably because of the way the eye works between the substrate, so-called Brooks membrane, and the pigmented cells, the black cells at the back of the eye, which is one reason why the elderly are subject to retinal detachment. That accumulation means that the pigmented cells don't stick as well. Essentially, you're trying to glue the pigmented cells onto a substrate that's covered in soap. It's not easy to glue something onto a soap-covered surface. And so that is, in fact, that basic result. Later on, there was actually a series of experiments that were done that validated that claim that there was a reduced adhesion that was causing this problem. We've applied this a little bit to the problem of diabetic retinopathy. Diabetic retinopathy affects the vessels on the front of the eye more than the back, the front of the retina rather than the back. One of the things that's done, not quite so much now, so this work is getting a little bit old perhaps, is to do laser ablation of the photoreceptors to try to reduce the oxygen burden on the retina and try to reduce the demand for oxygen. And that also reduces the level of growth factors. In this case, it's hypothesized that the problem is actually excess growth factors. You usually think of growth factors as maintaining cells, but if the growth factor levels become too high, the cells misbehave. And we were able to do a series of simulations of blood flow in the capillaries. These happen to be taken from an individual patient, although the network isn't very extensive. Predict what the oxygen level would be in the tissue, calculate then what the growth factor levels would be, and predict again the risk of a particular blood vessel causing a problem. And that would allow you, in principle, to target those laser ablations in regions of high risk to minimize damage. We've worked a little bit, actually, on polycystic kidney disease. This is a genetic disease that's caused by mutations in two genes, polycystin 1 and 2. It causes overgrowth of the kidneys. At the moment, the only treatment is kidney transplant. Most people with the disease die. It wasn't understood how these genes connect to the phenotype where you get overgrowth and cystic formation in the kidney. Our clinical collaborator, Bob Bacalhau, had an idea that it was associated with expression of a particular cell adhesion molecule, a defective cell adhesion molecule. And so we did a series of simulations of a small region of the kidney, a tubule, where we introduced a single cell with that gene mutation, which was represented as changes in the way the cell signaled and stuck to its neighbors. And we were able to reproduce the phenotype where you grow a cyst out of a single cell. That was then reproduced in vitro in cell culture lines of human kidney cells, where if you introduced this ectopic, inappropriate cadherin, you got a cystic phenotype. We then did some more classical bioinformatics with collaborators in Brazil who have some nice bioinformatics methods on this. And we're currently doing animal studies to see whether we have drugs that can modulate this kind of problem. We've worked a bit on toxicology over the years, in particular in the liver. In this case, we have a model of the distribution of molecular species in the whole body, PBPK models. Probably people have seen these in the course of this workshop. Instead, we've taken the liver component out and put in a very simple spatial model of one sinusoid. These are the equivalent of capillaries in the liver. The individual cells in the sinusoid interact with the blood. Here the blood flow is modeled explicitly. A chemical in the blood is transferred into the hepatocytes, those cells in the liver. They metabolize that chemical. In this case, we're modeling paracetamol. In the US, it's called Tylenol or acetaminophen. That molecule by itself is not a problem, at least not a major problem. But some cells in your liver metabolize it into a molecule called NAPQI, NAPQI, that's extremely toxic. And the result of that can be that liver cells die. And if you overdose on paracetamol, you can die very rapidly of liver failure. And so this was a model where we calibrated both the whole body model and the subcellular model to reproduce in detail the clinically measured levels of both acetaminophen and its metabolites that are measured in the blood. And so this is an example where we have actually a model of a drug being given to a patient, how it gets to the tissue, what it does in the tissue, and then how it's returned to the blood for a clinically measurable result. With a model like that, you can generate a virtual patient population and ask the question, within normal variation of the parameters in the model, are there outliers that could be either particularly susceptible or particularly resistant to toxins? And in this case, you're able to show that even small changes in individual parameters in metabolism and transport can lead to very large changes in the toxicity of that drug. That's important because it means that under some conditions, some people could be poisoned even by clinical levels of that drug. We've also done some modeling and experiments related to blood flow in the liver. The liver has a very complex blood flow. Blood flow direction actually reverses periodically in some parts of the liver. And again, you can use this kind of simulation to model how that changes the distribution of exposure of the cells in the liver to a particular molecular species. Most recently, and this is how Rahman and I met, we've been working on modeling infection in tissue patches. There are many, many questions that are still unanswered, although many of them are being answered in the past year, about what happens when you have a viral infection. We started out building a very simple model of a patch of epithelial tissue. This could be in the lung. We imagine that that patch is infected by a virus. The virus will begin to reproduce and spread in the tissue. At the same time, there will be chemical signals, cytokines, that signal the immune response in the tissue. The immune response calls in immune cells. Those immune cells kill infected cells. And so there's a race between the spread of the virus and the rate at which the immune cells clean up the infected cells. Unfortunately, in humans at least, once a cell is infected with a virus, typically the only way to get rid of it is to kill the cell. Here we've built a model where we have epithelial cells and what they're doing. We have a detailed subcellular model of viral replication. Those cells send out chemical signals. There's also a network model of the production of new immune cells in the lymph node and their transport back. The immune cells themselves move around. They kill target cells. They respond to chemical fields. So it's quite a multi-layer model. If people want to try this online, it's available at nanohub.org, tools CC3D COVID-19. And you can run this simulation if you're bored on your laptop while we're talking. This initial model wasn't a very detailed one, but we wanted to see what you could do with it. We wanted to model an example of the first drug that was out there for COVID. This was back a little over a year and a half ago now. Remdesivir. Remdesivir blocks the RNA replication phase of the viral life cycle. We have a model using SBML in the form of antimony of viral replication inside each cell. So the replication of each cell can be controlled by the level of the drug, which is supplied externally. So every cell can behave differently in terms of viral replication. We can simulate in that base simulation the effect of adding a drug that inhibits viral replication at that particular point of RNA. And we can then have higher dosage going to the top here and ask the question, during the infection, you don't usually know you're infected immediately. We can change the time of first treatment. And it's known already that the problem with antivirals is that typically by the time you feel sick, it's too late for the antiviral to work. If you take the antiviral before you get sick or immediately upon getting sick, the antivirals work very well. But if you wait until you've gotten sick, gone to the doctor, gotten the prescription and take it, usually it's too late. And so here on the top, left to right, left is early, right is later in the infection. Green means the infection was controlled. Red means it wasn't. White means that sometimes it was controlled and sometimes it wasn't. There's a lot of stochasticity in the response. And we were able then to do some basic simulations of the trade-offs between dose and timing in this kind of drug. That was really a proof of concept. We've gone back and tried to model the pharmacokinetics, a little bit more detail of remdesivir. Remdesivir is a prodrug. It's given by injection. These days it's really being phased out in favor of orally available drugs. But that means that the availability of the drug oscillates because it's not given all the time. It's given once per day. And so the level goes down between doses. And it takes time for it to build up initially. And that leads to a sort of an interesting barrier between the region where the drug is effective and the region where it isn't. And you can then study the effects of stochasticity in the system. In this case, the stochasticity makes things worse. If some individual cells don't get a high enough dose of the drug, they'll produce virus even if most of the tissue is not, and that can cause a problem. We've been working a little bit on early antiviral resistance as well. In this case, we're modeling so-called interferon pathways and trying to understand how they behave. Here, modeling the spread of virus, not in human, but rather in a cell culture well, and how that can be controlled by the use of interferon. Those models all had a very simplified understanding of the way the immune system worked. One of the things that we can do relatively systematically now is take the ODE models that you might have worked on earlier in this workshop and translate those into these kinds of spatial models. And so this was a paper that we just published. It's, I think, officially still in electronic only form in Biophysical Journal, modeling Journal of Theoretical Biology, modeling the spread of influenza in a tissue. In this case, there were detailed models, detailed experimental data available on not just the viral load, but on many of the chemical signals and how they changed in time, and also the particular immune cell species and how they changed in time. And there was a beautiful model by Price and Mohan et al. From JTB about 5 years ago that built an ordinary differential equation model, a reaction kinetic model of these species. We translated that into a cellular model where we have explicitly modeled macrophages, natural killer cells, CD8 plus T cells, CD4 plus T cells, B cells, antigen presenting cells, and many of the chemical species to try to understand both spatially and temporally how the immune system responds to influenza. In particular, in this case, as you change the initial dose of infection, you're exposed to the low level of influenza, you survive. Higher levels, the influenza outruns the immune system and can lead to death, and you're able to replicate that in this kind of simulation. I'll just mention Mechanica. Mechanica is a new software package that we're developing at the moment. It's just starting out to try to build these kinds of models on a larger scale. CompuCell can typically model a few thousand cells, 10,000 cells, 100,000 cells on a big computer. Mechanica is trying to model hundreds of thousands to millions of cells in a new way. So that's something that we're actively involved with. We're always looking for opportunities to work with people. They can be, these days with the internet, you can work with people anywhere. As I mentioned, the software is available to use online, so you don't even have to download it. It's all open source. But if people are interested in modeling any of these problems, one of the things we do a lot of is teach this kind of modeling and help people do it. We have biophysical models of cancer, vasculature, embryonic development, toxicity. I was talking the other day, I met somebody on LinkedIn who had been using CompuCell in his thesis in India. He called me, I think initially because he was looking for a postdoc, but it wound up he was hired by a company in India and went to work for them. But he actually was involved with a startup company in Singapore that was doing cancer modeling using CompuCell. And it was wonderful to hear because we didn't know about it. And that's a huge success. When you write software, you hear complaints all the time about the software. So when things don't work, we know about it. So when a company gets started using your software and you never hear from them, it means they were actually able to use it successfully. So we are very happy about that. We're always interested in people who want to work on these problems. As I mentioned, my lab has some opportunities. There are others also who have opportunities. And so if you're interested, please check it out. There's a good question, which I will come back to in just 1 second. Let me just finish that, my one thought, and then I'll come back to that, because that's actually a hard question. Rahman already mentioned, we have this IMAG MSM discussion group, Multiscale Modeling in Viral Pandemics. A lot of what they talk about is mechanistic modeling. The kind of modeling that I'm talking about here is only a very small portion of that. There's much more of the kind of reaction kinetic modeling that I think you've been hearing about more, but you'll certainly hear the occasional talk on multicellular modeling, both using our framework and using the many other frameworks that are out there. These days, there are quite a few. There was a good question here about sample parameters for virtual population modeling. Are there established methods? And I think this is one of the biggest problems we face in all of these mechanistic modeling frameworks, which is if you're going to do models of chemical reactions, you have rate equations. Those rate equations at least will have a velocity of the reaction if it's mass action. And typically you'll also have something like a Michaelis constant or a Hill constant and possibly a Hill exponent. Measuring those rate constants is not easy. And so there's a lot of uncertainty when you build network models about what the rate constants are. When they're measured in experiment, very often those conditions under which those rate constants are measured aren't the ones that are relevant in a particular in vivo situation. And so you really do have a problem of overfitting in these models. One of the things that helps you is that biological systems are very robust. And I joke with my students that if changing any parameter by a factor of 5 makes a big difference, the model design is wrong. Now, clearly, when you're talking about dosing for a drug, that's not the case. A change of 5 in the drug dose should make a difference, but it's not. It's an issue. If you're thinking about virtual populations, that's a little bit different. The problem with virtual populations is that typically parameters don't vary independently of each other in humans. There'll be correlations between parameter values, so that if there's a change in the rate of one particular rate law, there'll be changes in others. And that's hard to account for. In our virtual populations, typically we don't do anything fancy. We'll pick, say, a Gaussian distribution over 30 or 40 parameters, and we'll explore that parameter space. And the hope is that you're exploring the range of possibilities. However, if you look at a company like Nova Discovery, which is doing virtual clinical trials, they are developing technologies to actually try to build more systematic explorations of realistic variations in patient parameters. And so I think that's a great question. I'd love to talk about it. I'm not an expert in it. One of the postdocs in my extended group, Tarun, who's now going to be going off and working for a pharma company, his specialty is in trying to develop virtual patient populations. And so I'm sorry about not having a good answer for that. I think it's an open question that's definitely worth working on now. I would say just my last comment was, if there are any people in the room who are software engineers, there are lots of opportunities to do software engineering in the world of this kind of modeling. Our software is designed as open source software built around DLLs, so people can build components that can be integrated independently. And so if people are interested in those kind of problems, please come talk to us. We're always delighted when people extend our core software. And so I hope that gives you some idea of some of the capabilities of this kind of multicellular or virtual tissue modeling. There are a lot of things that one can do with it, many things beyond what I've talked about. But I hope it gives you a little bit of a flavor for that. We teach our annual course. The videos, they're rather tedious to watch that many hours of video online, but they're there on YouTube from last year. We're currently trying to organize our course for this coming year. We will probably do it virtually again because while there are advantages to doing it in person, wonderful thing last year was we had participants from all over the world. There were a number of people from India who stayed up all night to take the course. We were very grateful to them for doing that. We had participants from China, from Australia, from Africa, from South America. And when we did it in person in Bloomington, Indiana, unfortunately, many people couldn't come. It was too expensive. The trip was too hard. And so we really felt that it was important to do it virtually. I would love to have the opportunity to come to India and do this course in person at some point, because I would love to be able to bring these methods to India. Over the years, we've taught versions of the course in France, taught it in Brazil once, we've taught it in China once. But these days, again, with virtualization, you can get a lot of the benefit by doing it online. Again, if people want to try our code out, you can get it download or online. My email is jaglazier@gmail.com. If you want to find out more about our university and programs, lottieindiana.edu is the school. Engineering.indiana.edu is our department. Always looking for students. And I want to tell you about the new Embryo Institute at Purdue. Saturday, they have their kickoff meeting. I think they have 20 or so student and postdoctoral positions open at the moment at Purdue for this project. Again, the kind of my modeling is only a tiny piece of it. They have many, many mechanistic modelers of all kinds. We're looking for people. Acknowledge my funding from the U.S. National Institutes of Health, from the National Science Foundation, from Indiana University, and because through my pension plan, I own a little bit of Gilead stock, not much, and because I mentioned remdesivir, I have to disclose that I own shares in Gilead. Thank you very much for your patience. I really appreciate you being here today.
Thanks a lot, Dr. Glazier. It was a wonderful talk. I think the participants really enjoyed it. So there are a few questions in the chat box, but I'll hand you, I'll just ask Himanshu to unmute and speak. Himanshu?
Hi, James. Wonderful talk. Yeah. So I am sort of intrigued at the kind of framework that you're developing, especially the virtual population framework. So while you say that you can change the parameters by a factor of 5, and these are like drug concentrations, is it also possible to change, let's say, network edges in which the gene network is interacting or gene environment interactions that are happening? Because there is a lot of data that is already there where tissue expression profiles have been done in multiple, like single cell expression profiles have been done in multiple tissues. And we can sort of start maybe applying some of the principles and techniques that you're developing on those data sets to understand which are the important edges that get perturbed as the population changes or the environment changes. So any ideas on that?
So one thing that one could spend many lectures on is the difference, how to understand the difference between structural differences in networks and parametric differences in networks. At some level, you could imagine that you have your chemical species and every possible interaction is there and they're turned off. That would be a little bit like doing a neural network where you have all-to-all connections. So in principle, there isn't fundamentally a difference between parametric differences and structural differences if you imagine a network where everything possible could happen. Of course, practically, we do make that distinction. We have a network which has certain interactions and not others. And so trying to model the differences in structural effects of networks is something that's very hard, and typically things like optimizers don't work well, or for that matter, backpropagation in networks doesn't work well, because everything depends on the differentiability of your cost function. And so if you have a discontinuous change because you add something that wasn't there before, those algorithms tend to fail. So one thing that is certainly very interesting, and I'm trying to get a student to work on now, would be an example of that, where a classic question, which is now going to be quite important as we unlock, for example, in the United States, would be co-infection. If you're infected with influenza and COVID, and you can build a simple model where you have the virus compete for cells. You say the cell can only be infected by one or the other, and you infect with one first and the other. And the result isn't very exciting. The one that you give first typically wins because it gets more real estate. But in fact, we know that some respiratory infections like RSV send out interferon signals, promote interferon signals that then promote antiviral resistance, which means that then it's harder for the influenza to get a foothold. Whereas influenza inhibits interferon signaling, and that means that it's easier for RSV. And so if you get influenza first, the RSV is likely to be worse. Whereas if you get RSV first, you're likely to have the influenza case be mild. And so you can then build a series of structural perturbations where you say, what are all the possible interactions between these two signals, these two viruses, between cells? You can do it inside of the cell too. But here we would be asking the question, is that inhibition or activation happening not within a single cell, but between cells? And there you have enough, it's a simple enough set of possible interactions. The combinatorics don't kill you. And so you could, in principle, then do every possible combination and ask, how then parametrically are these different structural combinations different? Was that addressing what you were asking a little bit? Or did I misunderstand the graph?
So I was actually,
Understand the question?
No, I think you are right. So what I was thinking in terms of, let's say, when we are talking about populations, right? Usually when we say a genetic network, we apply what is called an additive model, and we identify QTLs, and we say these are the markers that are important for the phenotype. And on some studies would go on to say, okay, these markers interact to enhance the phenotype or enhance the risk, or whatever you want to say, right? So now, given that is the case, if we now perturb, so what I'm trying to understand is that can we take structural perturbations across interacting nodes and change the degree of interaction, right? Because some mutations in QTLs, for example, can change the strength of interaction. So what you're saying, the dosing, now you're changing the strength. So, and that would be a smaller set, because I'm not talking about 30,000 genes. I'm talking about, let's say, 10, 15, maximum 25, 30 loci. So is that a computationally feasible process, or still would be harder?
So one of the people who spoke early on in our seminar series, in our working group series, was somebody named Kevin James at University of Virginia. And he was doing a series of very beautiful experiments on viral infection, trying to map quite specifically the effect of specific genetic variations on the immune response. And he was able, in that particular case, to get a quantitative relationship between particular affinities due to particular gene mutations and the phenotypic outcome. That's, I think, path-breaking work, and it was very difficult. But I don't know anybody who's put it into this multicellular context yet. There is a group in Poland that's been working on this that I thought was very good. The lead of that group gave a talk in our working group, but not on that topic. We hoped he'd talk about that. He talked about something else. But I think that's just in its infancy. But yes, I think this modeling could do that. The key is, of course, to connect the genetic variation with some numerical parameter in the problem.
Yes.
One place this comes up in a big way is in cancer, because people think of cancer as associated with gene mutations, but a mutation could increase the rate at which something happens or decrease it. So cancer is really not about mutation, which is a source of variation. It's about selection.
Yeah.
And there's been, nowadays, people talk a lot about somatic evolution and about microenvironmental effects on cancer. But that's new. 20 years ago, people didn't think about that.
20 years ago, people.
And people like Bob Gatenby at Moffitt in Tampa, Florida, were pioneers. Gatenby was thought of as a bit of a crackpot for decades, talking about somatic evolution and microenvironmental effects in cancer. People didn't listen because they wanted to think everything was genetic, not about selection. And he was right. And he's built probably the best computational cancer group in the world now in Tampa. At Moffitt, they have Sandy Anderson, they have David Basanta, they have Keiko Enderling, they have Kasia Resniak. They have a tremendous group of people doing this kind of modeling. They use their own package, unfortunately, not Capusel, but that's fine. But doing this kind of modeling and turning it into clinical practice. They're improving therapies for prostate cancer and for many other kinds of cancer using exactly the kinds of things you're talking about. Okay. So I would encourage you to look at their work.
Okay, great. Thanks.
Especially on prostate cancer.
That's fine. Like the principle remains the same. You can start applying. Okay, great. Thanks, James.
There's one more question from Kandapiran. He asks, how does a model scale from one level to another? Are crucial or key parameters identified well ahead and executed, or simultaneous dynamic models generated at various levels with some threshold as a trigger? So this is in the chat box if you want to look at it.
I apologize. I was looking at the question and answer box, but not the chat box during the talk. And so I missed some of the questions. So I think that this is also something that is difficult. Modern molecular biology is tremendously good at measuring molecular concentrations. It's very hard, however, to connect a specific molecular concentration to a cell behavior. You could have two cells that are molecularly identical that are quite different in their behavior. Now, there are signals, so changes in levels make a difference. If you see activation of p53, you're probably going to have a cell that's going to die through apoptosis. If you see activation of certain molecular species, PIP2, PIP3, MAP kinase, RAS, you may see cell motility increases or cell cycle increases. But I think that the question about how you, and that maybe I'm missing the question a little bit, but the question about how you interconnect levels at a quantitative level is still something that is not fully understood. And I think that this is a problem that's intrinsic to biology. It's one of the reasons biology is very hard. The second question really was, do we know in advance what these connections are going to be? And you say, parameters identify well ahead and execute our simultaneous dynamic model generate. We typically will generate alternative models. So for example, we can't generate all of them because the combinatorics is impossible. You can have an infinite number of possible model structures. But as an example with that, going back to that somite example where we had the molecular clock, that molecular clock was published based on published work of Goldbetter and Pourquier, who built a model of the clock inside a single cell. And when we implemented that model, which they had validated pretty well against detailed experiments, when we implemented that model within cells and coupled the cells, we realized that the model could not be right because there was no way for those oscillators to synchronize between neighboring cells. The model structure did not allow synchronization. And since that synchronization was observed, it meant that there had to be a structural defect in the model. And we didn't do a comprehensive exploration, but we did explore dozens of different model structures to ask which possible model structures would allow the synchronization that was observed. And that was something where if you studied what happened inside a single cell, there was no way to know that there was a problem, because the model inside the single cell reproduced the experiment fine. It was only when you asked the question, did the cells, when they interact, behave properly, that you were able to see that there was a problem with the model structure. There's a question on transport. So I guess it depends here on what you mean by transport. So diffusion of molecular species is built into CompuCell. So diffusive transport is there. One of the mathematical complexities of biology is that you have interaction of short-range and long-range signaling mechanisms. In particular, you have active transport, advection with the blood, and that carries signals over long distances. In some of the models we'll use, the blood is modeled essentially as a short circuit, where it creates a boundary condition at the surface of blood vessels, for example, for oxygen. And that's relatively easy to do. In other cases, you want to do an explicit model of transport in the blood. And in that model of the sinusoid, in the liver sinusoid, we explicitly modeled the blood flow. We basically did a flow model of blood in the sinusoid. The problem with doing that is that blood flow is fast. And so then the time scale of the time steps in your simulation goes from being a minute or two to being milliseconds. And that means your simulation slows down by a factor of 1,000. And the reason that simulation only had one sinusoid instead of the complication of real blood flow in the liver and the architecture in the liver was because that blood calculation was so expensive. In later simulations, we did more of a bucket brigade Kirchhoff-like solver to do the blood flow. But one of the things, that we did by hand. We wrote that in Python and put it in as a Python module. I would love to have an integrated blood flow model inside of CompuCell. We've looked into trying to integrate solver packages that do advection very well. There's an open source package called Phoenix. It's very good for solving flow models. And that integration in principle is possible, but there were some issues with basically with the Phoenix side of things that made that difficult. There are people who have tried to integrate CompuCell with some other open source packages. In fact, the student I mentioned from India who came to talk to me a few weeks ago, he had been integrating with a Python wrapped finite element solver, solid solver, and that works well. Anything that has a Python interface you can use. Building it inside so that the primitive objects are available is a little more work, but that would be something, if somebody wanted to do that, we'd love to see that. The Mechanica framework, because it's particle-based, allows particle-based fluid solving simultaneously. And so that was designed from the ground up to allow you to do advection and transport using the same framework that you're using to do doing the cells. And so there's still pieces of that we're working on. But one of the reasons we designed that new framework was specifically to allow that to work. But diffusive transport exists. Advective transport exists in CompuCell, and people use it, but it's computationally expensive. So it means either your simulations take a long time to run or you have to do small simulations. Okay. I know it's getting late for you there, so I appreciate you being willing to stay after hours for this.
So there are also a couple of questions in the Q&A. So, for example, are there any limitations in the kind of drugs that can be tested for response?
Well, the only thing that you need is you have to have some idea of the biological mechanisms at work. And ideally, you'd like to know where the drug goes and how its ADME, its availability, dosimetry, metabolism, and elimination work. So if you have those standard pharmacokinetic parameters, and you have some idea of what the drug does, there's no magic. I mean, when I talk to clinicians, they often think modeling can do nothing, or they think modeling is magic. And if you explain what you need to know to be able to build a model, I'll often come back and say, if I knew that, I wouldn't need the model. So that's true with drugs as well. In the case of remdesivir, the reason we picked it was because there was, in fact, a parameter already in our subcellular viral replication model, our network model of viral replication, that was the rate at which the RNA was replicated. And there were experiments that measured, as a function of drug dose, the rate of RNA replication. And so in that case, there was a direct measurement, experimental measurement, of the relationship between the drug and a parameter that we had in our model. So it made it very easy to model. That's why we picked that. In the case of the liver, the particular molecular pathways that cause that particular toxic event are understood. But the level of the molecular species, the particular cytochrome that causes that problem, is regulated. And that regulation is not well understood. So the origin of the variation between the hepatocytes is not well understood. It's been measured, but why it's got that distribution is still debated. And a problem in that liver model is that the level of the cytochrome P450s, in particular CYP2E1, is a problematic one. That level of cytochromes changes in the liver because in response to what you eat, what drugs you take, whether you've had alcohol, your liver changes its metabolism. And how that metabolism is regulated is not well understood. It's a very hard problem and something that needs to be understood if we're ever going to really get dosimetry to work in detail. That's a big unsolved problem at the molecular biology level of how the liver works. But when it's measured, you can certainly put it in. And there was a question, which is, how is multiscale modeling different from single-scale modeling? Well, I hope the talk answered some of that in the sense that what we've looked at, and I didn't go into any detail on any of these problems, but I have talked about a couple of cases where there was an existing model of what was going on inside of a single cell. There was a model of how a virus reproduced inside a single cell. There was a model of how interferon signaling happened in a single cell. There was a model of how chemicals oscillated inside of a single presemitic mesoderm cell during development. When you put those cells in context, which makes it multiscale, everything changes. And so I hope that this gives you a sense of you need to look inside a single cell to understand what it's doing. But no amount of looking, even if we knew everything about what happened inside a single cell, if we didn't model how the cells interacted, we wouldn't be able to understand how tissues form and fail. It's a little bit like the issue of driving a car. If you drive a car by yourself, you know how the car behaves. If you're stuck in traffic, and from my experience the one time I was in India, I remember that being stuck in traffic was an experience I have it here too, but maybe more there. the traffic jam is not a property of an individual car. It's a property of an ensemble of cars interacting with each other. And tissues in life aren't by themselves a property of what's happening inside of a single cell. They're property of what's happening with an ensemble of cells in a context and how their control interacts. And so I think that you need the subcellular modeling very much, but to really go forward, you need to be able to put that into a multicellular context.
Let me ask one question. Okay.
Please.
Okay.
Go ahead.
So I think it's late, but yeah. So I think, I mean, thanks for the exciting and interesting talk, James. So, you know, morphogenesis is one of the fields which has a long way to go. We still don't know how from the embryo every single organs and parts arise, yeah, and definitely there is a scope that modeling can help. So what do you think, what needs to improve further, the molecular biology, what we know, or the modeling framework itself, so that we can learn more about morphogenesis? Do you think where is the gap?
Well, I'd say that, I mean, the progress of molecular biology over the past 30 years is amazing. You can measure every concentration of every molecular species inside a cell with tremendous accuracy. Not so much while the cell's alive, but if you're willing to kill it, you can do single cell proteomics, which is amazing. On the other hand, there are a lot of things that are very hard to measure. We can't measure the forces cells exert on each other. We know relatively little about how the cytoskeleton works at a force-based and structural level. The molecular species controlling how cells move are understood. There's more and more biophysics about how cells exert forces. But if I ask the question, if I give drug X to cell Y, is the cell going to crawl faster or slower? That's still hard to predict because the macromolecular machines inside of cells are so complex. And proteomics treats the cell as a bag of chemicals. It neglects the structure inside cells, and where things are and how they're organized matters. And that's true at the tissue level as well. The other thing that we understand still very little about is extracellular matrix. We think of the genome as storing information, and it does. But in an adult, or even in a developing embryo, that information is translated into structure, extracellular matrix structure. And that extracellular matrix has stored information in it that we have very little access to. We don't understand the forces in extracellular matrix. We don't understand the chemical signals in extracellular matrix. And a classic example of this that demonstrates it would be in tissue engineering, where you take a tissue from an animal, you kill the cells, you wash out all of the cells, and then you put new cells in, and they go back to where they would have been in the original tissue because there's signals in the extracellular matrix that give instructions for where cells need to be. Some of those are chemicals bound to the ECM. Some of those are the mechanics and the architecture, the fiber organization of the ECM. During embryogenesis or wound healing, the fibers in the extracellular matrix are reorganized. We don't understand how ECM fibers are generated. We don't understand how they're reorganized, and we don't have a good way of modeling it. My old student, who's now on the faculty at North Carolina State, Giulio Belmonte, has been doing some beautiful work trying to model fibers and how they work. And I'd say fiber architecture and ECM and understanding it both at the experimental level and modeling level is just at its infancy and is very important. And so I think there are big gaps in our understanding both of the basic biology, the biophysics, and our ability to model. These are very hard problems.
Thank you.
Okay, so thanks a lot. Dr. James, for your time. Really appreciate it early in the morning. And I think the participants truly enjoyed it. It's been a grueling 4 days of our winter school. And I think, they've all been here and many of them have participated. And yeah, there are lots of comments on, they enjoyed the talk. Thank you so much.
Okay, thank you. And I hope I'll be hearing from you again, and look forward to hearing more. And
we would love to host you sometime for a, for one of these
meeting, right?
workshops. Yeah, in person or virtual, right? So these hackathons, and because lots of students who are excited about these kinds of things here, so it would be wonderful. Yeah.
Okay, well, I'd love to come again. It would be fun, and look forward to hearing from you. And try CompuCell out. Tell us if it works. Wonderful. If you don't like it and it needs to be fixed, tell us, and either we'll fix it or maybe we can have you fix it for us. Okay. Thank you, everybody. I appreciate it so much.
Thank you. Bye.